Welcome to this explainer. We've designed this specifically as a high-yield study guide for medical students and future clinicians. Today we're going to break down the bacterial growth curve. And we aren't just looking at this as some simple laboratory phenomenon you memorize and forget. We're looking at it as a dynamic, high-yield clinical battlefield. Understanding exactly how a bacterial population grows, adapts, and ultimately dies is absolutely foundational to understanding pathogenesis. More importantly, it tells us exactly when to strike with antimicrobial therapies.
Okay, let's dive into this framework. We'll be mapping out the pathogen's progression through the infection timeline, the lag phase, the log phase, the stationary phase, and finally, the decline phase. All right, let's kick things off with Section 1, the infection timeline, and look at how we actually measure this microscopic battlefield.
To really understand this battlefield, we have to look at generation time. That's simply the sheer speed of bacterial division. This baseline metric dictates the entire tempo of an infection, and it brilliantly illustrates the clinical stakes. On one extreme, you have the hyper-aggressive Clostridium perfringens, which has a remarkably fast generation time of just 10 minutes. 10 minutes. On the other side, you've got the slow, insidious Mycobacterium tuberculosis, which takes a grueling 12 to 16 hours to do the exact same thing. Clinically, this is huge. It means a Clostridium infection can rapidly overwhelm your patient in a matter of hours, making it an acute medical emergency, right? Whereas tuberculosis, that establishes itself through a prolonged chronic campaign.
Now, to actually track these infections and figure out that specific generation time, scientists use this classic, high-yield, four-step laboratory methodology to build the bacterial growth curve. They start by growing a pure culture in a liquid medium at a constant, highly controlled temperature. Then they collect samples at fixed intervals, say maybe every 30 minutes. Next, they determine the number of viable living organisms in each sample. And finally, they plot the logarithm of the number of bacteria per milliliter against time. This is what gives us our definitive visual map of the bacteria's entire life cycle.
Let's pause for a second and memorize this crucial summary table. It's really your master blueprint for the rest of this explainer. Look at how the growth rate goes from 0, peaks at a constant maximum, flatlines back to 0, and then ultimately turns negative. Meanwhile, the cell's metabolic priorities are dramatically shifting across these phases. They go from enzyme activation to rapid cell division, then to defensive toxin production, and finally to self-destruction.
Let's look closer at the first step in that blueprint. Section 2, the lag phase, which is all about tactical preparation.
What's genuinely fascinating here is that the newly inoculated bacteria aren't actually multiplying yet. The population growth rate is 0. Instead, during this tactical lag phase, the pathogen is essentially casing the joint. It's assessing the host environment. It's furiously booting up its enzymatic machinery, synthesizing RNA and grabbing growth factors. Oddly enough, the individual bacterial cells are actually increasing in physical size, but there's no appreciable increase in their numbers. Depending on the species and conditions, this phase can last an hour, or it could last several days. Basically, they are literally gearing up for war.
Once they're fully prepared, the pathogen launches its blitzkrieg. Section 3, the log phase, an exponential attack.
Here, unchecked binary fission leads to maximum division. The population is growing exponentially. One cell becomes 2, then 4, 8, 16, you get the idea. The microbes are multiplying at their absolute maximum possible rate. This makes it the phase where bacteria are the healthiest. If you were to look at them under a microscope right now, the cells would appear small and uniformly stained. And because we're plotting this on logarithmic graph paper, this exponential attack charts perfectly as a steeply sloped straight line.
So, what does this all mean for you clinically? Well, this is absolutely crucial. Because the bacteria are rapidly dividing right now, their cellular machinery is working in massive overdrive, and that creates the exact window of maximum vulnerability. Bacteria in the rapid log phase are at their most sensitive to adverse conditions. This is the optimal time for your antibiotics and antimicrobial agents to strike. You want to interrupt those highly active reproductive and synthetic processes. If you want to clear an infection efficiently, this is for sure the phase you want to target.
But as we know, this aggressive expansion eventually hits a wall. Welcome to Section 4, the stationary phase, a toxic stalemate.
That rapid exponential growth screeches to a complete halt. In this stationary phase, the death rate of the bacteria begins to exactly equal the replication rate, which totally flattens out the curve.
And this happens entirely due to a brutal progression of cellular stressors. Essential nutrients are completely exhausted, water and oxygen are depleted, the pH of the environment shifts and becomes hostile, and crucially, the bacteria are swimming in an accumulation of their own toxic metabolic waste products. They've reached peak population density, but the environment simply can't sustain them anymore.
Facing starvation and this massive toxic buildup, the bacteria don't just sit there waiting to die. They panic. Well, actually, scratch that. They don't panic. They desperately and highly specifically shift their gene expression. Gram standing becomes really variable and irregular here. Many bacteria begin to deploy heavy defenses, forming these highly resilient endospores to survive the famine. Simultaneously, they start producing and releasing deadly exotoxins. From a clinical perspective, this is a real game changer. Even though bacterial replication is stalled out, the patient can become incredibly sick during this specific phase, precisely because of this toxic waste and exotoxin production.
Eventually, the environment becomes completely lethal, dragging the pathogen into Section 5, the decline phase, leading to structural collapse.
The death rate now officially exceeds the replication rate. As the cells succumb to the sheer exhaustion of nutrients and the massive volume of their own autolytic enzymes and toxic products, we witness a total structural collapse. The bacteria begin to degrade into what are called involution forms. These are completely bizarre, aberrant shapes. We're talking swollen, long, filamentous rods or branching globular forms that become super difficult to identify under a microscope. Some even lose their cell walls entirely, becoming protoplasts, spheroplasts, or L-phase variants.
Now, looking at these twisted, wall-less remnants, you might assume these degraded pathogens are completely defeated. The infection's over, right? Time to celebrate? But let's ask a really vital clinical question to end our explainer today. Are these bizarre involution forms truly dead forever?
Astonishingly, no, there's no way. The research tells us that when these broken down, involuted forms are introduced into a fresh, nutrient-rich medium, they usually revert completely back to the original shape of the healthy bacteria. They can literally rise from the ashes. And that leaves us with a chilling clinical thought to ponder. How many seemingly cured infections, pushed right to the brink of the decline phase, are actually just harboring these dormant L-phase variants, just waiting for the right nutrients and host vulnerabilities to strike again? Thank you so much for joining us for this explainer, and definitely keep that in mind the next time you review a patient's chart.